Some cytokines of innate immunity, interleukin 1 functions. We will be learning the functions only. Okay. Vasculature, inflammation, hypothalamus, fever, liver, introduction of acute phase proteins. Then tumor necrosis factor alpha, TNF alpha, macrophages, vasculature, inflammation, liver, induction of acute phase proteins, loss of muscle, body fat, reduction of death in many cell types, neutrophil activation. Then interleukin 12, IL-12, macrophages, dendritic cells, NK cells, influences, adaptive immunity. Then interleukin 6, macrophages, epithelial cells, liver, induces acute phase proteins, influences, adaptive immunity, proliferation and antibody secretion of B cell lineage. Then interferon alpha, IFN alpha, macrophages, induces an antiviral state in most nucleated cells, increases MHC class 1 expression, activates NK cells. Then interferon beta, IFN beta, fibroblasts, induces an antiviral state in most nucleated cells, increased MHC cell 1 expression, activates NK cells. Some cytokines of adaptive immunity. Interleukin 2, T cells, T cell proliferation can promote AICD, NK cell activation and proliferation, B cell proliferation. Then interleukin 4, IL-4, Th2 cells, mast cells, promotes Th2 differentiation, isotype switch to IgE. Interleukin 5, IL-5, Th2 cells, eosinophil activation and generation. Interleukin 25, unknown, induces secretion of Th2 cytokine profile and transforming growth factor beta. T cells, macrophages, other cell types, inhibits T cell proliferation and effector functions, inhibits B cell proliferation, promotes isotype switching to IgE, inhibits macrophages. Interferons gamma, IFN gamma, activates macrophages, increase expression MHC class 1 and class 2 molecules, increase antigen presentation. In vivo, however, cytokines rarely, if ever, act alone. Instead, a target cell is exposed to a melanome containing a mixture of cytokines whose combined synergistic or antagonistic effects can have very different consequences. In addition, cytokines can often induce the synthesis of other cytokines, resulting in cascade of activity. So this we just mentioned. Then the non-specificity of a cytokine seemingly conflicts with the established specificity of the immune system. What keeps the non-specific cytokines from activating cells in a non-specific fashion during the immune response? One way in which specificity is maintained is by careful regulation of the expression of cytokine receptors on cells. Often, cytokine receptors are expressed on a cell only after that cell has interacted with the antigen. In this way, cytokine activation is limited to antigen activated lymphocytes. Another means of maintaining specificity may be a requirement for direct interaction between the cytokine producing cell and the target cell to trigger cytokine secretion, thus ensuring that effective concentrations of the cytokine are released only in the vicinity of the intended target. In the case of the Th cell, a major producer of cytokines, close cellular interaction occurs when the T cell receptor recognizes an antigen MHC complex on an appropriate antigen presenting cell, such as a macrophage, dendritic cell, or B lymphocyte. So, cytokines secreted at the junction of these interacting cells reach high enough local concentration to affect the target ABCs, but not more distant cells. In addition, the half-life of cytokines in the bloodstream or other extracellular fluids into which they are secreted is usually very short, ensuring that they act for only a limited period of time and thus over a short distance. Another means of maintaining specificity may be a requirement for direct interaction between the cytokine producing cell and the target cell to trigger cytokine secretion, thus ensuring that effective concentrations of the cytokine are released only in the vicinity of the intended target. In the case of Th cell, a major producer of cytokines, close cellular interaction occurs when the T cell receptor recognizes an antigen MHC complex on an appropriate antigen presenting cell such as a macrophage, dendritic cell, or B lymphocyte. Cytokines secreted at the junction of these interacting cells reach high enough local concentration to affect the target ABC, but not more distant cells. In addition, the half-life of cytokines in the bloodstream or other extracellular fluids into which they are secreted is usually very short, ensuring that they act for only a limited period of time and thus over a short distance. The next major topic is nothing else but the cytokine receptors. As noted already, to exert their biological effects, cytokines must first find specific receptors expressed on the membrane of responsive target cells. Because these receptors are expressed by many types of cells, cytokines can affect a diverse array of cells. Biochemical characterization of cytokine receptors initially progressed at a very slow pace because their levels in the membrane of responsive cell is quite low. As with the cytokines themselves, cloning of the genes encoding cytokine receptors has led to rapid advances in the identification and characterization of these receptors. So, receptor family, immunoglobulin subfamily receptors. Here we could see ligands IL-1M, CSF, C, KIT. Then cytokine receptors fall within 5 families. Receptors for the various cytokines are quite diverse structurally, but almost all belong to one of 5 families of receptor proteins. Immunoglobulin superfamily receptors. Class 1 cytokine receptor family, also known as the hematopoietin receptor family. Then class 2 cytokine receptor family, also known as the interferon receptor family. Then TNF receptor family, chemokine receptor family. Class 1 immunoglobulin superfamily receptors. Then class 1 cytokine receptors, that means hematopoietic conserved cysteines are there. Then class 2 cytokine receptors, interferons. Then TNF receptors, chemokine receptors. Many of the cytokine binding receptors that function in the immune and hematopoietic systems belong to the class 1 cytokine receptor family. The members of this receptor family have conserved amino acid sequence motifs in the extracellular domain, consisting of 4 positively conserved cysteine residues and a conserved sequence of tryptophan, serine, any amino acid, tryptophan, serine, where X is the non-conserved amino acid. The receptors for all the cytokines classified as hematopoietins belong to the class 1 cytokine receptor family, which also is called the hematopoietin receptor family. The class 2 cytokine receptors possess the conserved CCCC motifs but lack the WS motif present in class 1 cytokine receptors. Initially, only the 3 interferons, alpha, beta, and gamma, were thought to be ligands for these receptors. However, recent work has shown that the IL-10 receptor is also a member of this group. Another feature common to most of the hematopoietin class 1 cytokine and the class 2 cytokine receptor families is multiple subunits, often including one subunit that binds specific cytokine molecules and another that mediates signal transduction. Note, however, that these two functions are not always confined to one subunit or the other. Engagement of all of the class 1 and class 2 cytokine receptors studied to date have been shown to induce tyrosine phosphorylation of the receptor through the activity of protein tyrosine kinase closely associated with the cytosolic domain of the receptor. Then subfamilies of class 1 cytokine receptors have signaling subunits in common. Several subfamilies of class 1 cytokine receptors have been identified, with all the receptors in a subfamily having an identical signal transducing subunit. 3 receptor subfamilies named after GM, CSF, IL-2 and IL-6. The sharing of signal transduction subunits among receptors explains the redundancy and antagonism exhibited by some cytokines. Consider the GM-CSF receptor subfamily, which includes the receptors for IL-3, IL-5, and GM-CSF. Each of these cytokines bind to a unique low-affinity cytokine-specific receptor consisting of an alpha subunit only. All three low-affinity subunits can associate non-covalently with a common signal transducing beta subunit. The resulting dimeric receptor not only exhibits increased affinity for the cytokine but also can transduce a signal across the membrane after binding the cytokine. Interestingly, IL-3, IL-5 and GM-CSF exhibit considerable redundancy. IL-3 and GM-CSF both act upon hematopoietic stem cells and progenitor cells, activate monocytes and induce megakaryocyte differentiation. All three of these cytokines induce eosinophil proliferation and basophil degranulation with release of histamines. Since the receptors for IL-3, IL-5 and GM-CSF share a common signal transducing beta subunit, each of these cytokines would be expected to transduce a similar activation signal, accounting for the redundancy among their biological effects. In fact, all three cytokines induce the same patterns of protein phosphorylation. Furthermore, IL-3 and GM-CSF exhibit antagonism. IL-3 binding has been shown to be inhibited by GM-CSF, and conversely, binding of GM-CSF has been shown to be inhibited by IL-3. Since the signal transducing beta subunit is shared between the receptors for these two cytokines, their antagonism is due to competition for a limited number of beta subunits by the cytokine-specific alpha subunits of the receptors. A similar situation is found among the IL-6 receptor subfamily, which include the receptors for IL-6, IL-11, leukemia inhibitory factor LIF, oncostatin M OSM, and ciliary neurotrophic factor. So in this case, a common signal transducing subunit called the GP130 associates with one or two different cytokine specific subunits, LIF and OSM, which must share certain structural features. Both bind to the same alpha subunit. As expected, the cytokines that bind to receptors in this subfamily display overlapping biological affinities. IL-6, OSM and LIF induce synthesis of acute phase proteins by liver hepatocytes and differentiation of myeloid leukemia cells into macrophages. IL-6, LIF and CNT affect neuronal development, and IL-6, IL-11 and OSM stimulate megakaryocyte maturation and platelet production. The presence of GP130 in all receptors of the IL-6 subfamily explains under common signaling pathways as well as the binding competition for limited GP130 molecules that is observed among these cytokines. A third signal transducing subunit defines the IL-2 receptor subfamily, which includes receptors for IL-2, IL-4, IL-7, IL-9 and IL-15. The IL-2 and the IL-15 receptors are heterotrimers consisting of a cytokine-specific alpha chain and two chains, beta and gamma, responsible for signal transduction. The IL-2 receptor gamma chain functions as a signal transducing subunit in the other receptors in this subfamily, which are all dimers. Recently, it has been shown that congenital X-linked severe combined immunodeficiency (XSCID) results from a defect in the gamma chain gene, which maps to the X chromosome. The immunodeficiency is observed in this disorder due to the loss of all cytokine functions mediated by the IL-2 subfamily receptors. Then the IL-2R is one of the most thoroughly studied cytokine receptors. Because of the central role of IL-2 and its receptor in the clonal proliferation of T cells, the IL-2 receptors has received intensive study. As noted in the previous section, the complete trimeric receptor comprises three distinct subunits, alpha, beta and gamma chains. The beta and gamma chains belong to the class 1 cytokine receptor family, containing the characteristic CCCC and WX WSXWS motors, whereas the alpha cells has a quite different structure and is not a member of this receptor family. The IL-2 receptor occurs in three forms that exhibit different affinities for IL-2. The low affinity monomeric IL-2R alpha, the intermediate affinity dimeric IL-2R beta gamma, and the high affinity trimeric IL-2R alpha beta gamma. Because the alpha chain is expressed only by activated T cells, it is often referred to as the TAC, T cell activation antigen. A monoclonal antibody designated anti-TAC, which binds to the 55 kilodalton alpha chain, is often used to identify IL-2R alpha on cells. Interactions between cytokine-specific subunits and a common signal transducing subunit of cytokine receptors. Signal transduction by the IL-2 receptor require both the beta and gamma subchains, but only the trimeric receptor containing the alpha chain as well binds IL-2 with high affinity. Although the gamma chain appears to be constitutively expressed on both the lymphoid cells, expression of the alpha and beta chains is more restricted and is markedly enhanced after antigen has activated resting lymphocytes. This phenomenon ensures that only antigen-activated CD4 plus and CD8 plus T cells will express the high affinity IL-2 receptor and proliferate in response to physiologic levels of IL-2. Activated T cells express approximately 5 into 10 raised to 3 high affinity receptors and 10 times as many low affinity receptors. NK cells express the beta and gamma subunits constitutively accounting for their ability to bind IL-2 with an intermediate affinity and to be activated by IL-2. Then engaged cytokine receptors activate signaling pathways. While some important cytokine receptors lie outside the class 1 and class 2 families, the majority are included within these two families. As mentioned previously, class 1 and class 2 cytokine receptors lack signaling motors, intrinsic cytosine kinase domains. Yet early observations demonstrated that one of the first events after the interaction of a cytokine with one of these receptors is a series of protein tyrosine phosphorylations. While these results were initially puzzling, they were explained when a unifying model emerged from studies of the molecular events triggered by binding of interferon gamma, IFN gamma, to its receptor, a member of the class 2 family. IFN gamma was originally discovered because of its ability to induce cells to block or inhibit the replication of a wide variety of viruses. Antiviral activity is a property it shares with IFN alpha and IFN beta. However, unlike these other interferons, IFN alpha plays a central role in many new immunoregulatory processes, including the regulation of mononuclear phagocytes, B cell switching to certain IgG cells classes, and the support or inhibition of the development of Th cell subsets. The discovery of the major signaling pathway invoked by interaction of IFN gamma and its receptor lead to the realization that signal transduction through most, if not all, class 1 and class 2 cytokine receptors involves the following steps, which are the basis of unifying signaling model. The cytokine receptor is composed of separate subunits, an alpha chain required for cytokine binding and for signal transduction, and a beta chain necessary for signaling but with only a minor role in binding. The cytokine receptor is composed of separate subunits. Then different inactive protein tyrosine kinases are associated with different subunits of the receptor. The alpha chain of the receptor is associated with a novel family of protein tyrosine kinase, the Janus kinase, JAK family. The association of JAK and the receptor subunit occurs spontaneously and does not require the binding of cytokine. However, in the absence of cytokine, JAK's lack protein tyrosine kinase activity. So cytokine binding induces the association of the two separate cytokine receptor subunits and activation of the receptor associated JAKs. The ability of IFN gamma, which binds to class 2 cytokine receptors to bring about the association of the ligand binding chains of its receptor, has been directly demonstrated by X-ray crystallography. Activated JAK create docking sites for the STAT transcription factors by phosphorylation of specific tyrosine residues on cytokine receptor subunits. Once receptor associated JAKs are activated, they phosphorylate specific tyrosines into receptor subunits of the complex. Members of a family of transcription factors known as STATs, signal transducers and activators of transcription, bind to this phosphorylated tyrosine residues. Specific STATs play essential roles in the signaling pathway of a wide variety of cytokines. The binding of STATs to receptor subunits is mediated by the joining of this SH2 domain on the STAT with the docking site created by the JAK-mediated phosphorylation of a particular tyrosine on receptor subunits. After undergoing JAK-mediated phosphorylation, STAT transcription factors translocate from receptor docking sites at the membrane to the nucleus, where they initiate the transcription of specific genes. While docked to receptor subunits, STATs undergo JAK-catalyzed phosphorylation of a key tyrosine. This is followed by the dissociation of the STATs from the receptor subunits and their dimerization. The STATs dimers then translocate into the nucleus and induce the expression of genes containing appropriate regulatory sequences in their promoter regions. In addition to the IFN gamma, a number of other class 1 and class 2 ligands have been shown to cause dimerization of their receptors. An important element of cytokine specificity derives from the exquisite specificity of the match between cytokines and their receptors. Another aspect of cytokine specificity is that each particular cytokine or group of redundant cytokine induces transcription of a specific subset of genes in a given cell type. The resulting gene products then mediate the various effects typical of that cytokine. The specificity of cytokine effects is then transferable to three factors. First, particular cytokine receptors start particular JAK-STAT pathways. Second, the transcription activity of activated STATs is specific because a particular STAT homodimer or heterodimer will only recognize certain sequence motifs and thus can interact only with the promoters of certain genes. Third, only those target genes whose expression is permitted by a particular cell type can be activated within a variety of cells. That is, in any given cell type, only a subset of the potential target genes of a particular STAT may be permitted expression. For example, IL-4 induces one set of genes in T cells, another in B cells, and yet a third in eosinophils.